Welcome to our presentation for the investigation on antibacterial properties of flavonoid concentration of different liquors against E. coli or Escherichia coli and Staphylococcus aureus. This is prepared by Cherry Sakabagan, yours truly, and Anjali Sarabia, BI 303, Section 709 by under Professor Nurul Hassan Mohammed. Flavonoids are polyphenolic compounds primarily derived from plants, known for their significant biomedical interest. These natural pigments, including anthocyanins, give flower their red, purple, and blue hues, as are predominantly found in the outer skin of berries and berry-based products like wine. Flavonoids offer a range of potential health benefits such as antioxidant and anti-inflammatory actions along with the antiviral and anti-cancer properties. Particularly notable is their microbial potential, which has garnered substantial interest in some scientific communities. The significance of studying antibacterial properties in liquors. Liquors, especially wines, derived from plant materials are rich in flavonoids, making them important subject for antimicrobial research. The reach of flavonoid profiles position these beverages as potential reservoir for exploring new antimicrobial agents.
Objectives of our research to broadly evaluate the antimicrobial activities of various liquors such as red, white, and rice wine. We also include to investigate the role of flavonoids concentration in determining the antimicrobial strength of different wines, particularly well-known pathogens such as E. coli and Staph aureus. The last objective of our research is to understand the possible mechanisms by which flavonoids might exert their antibacterial effect, offering insights into their pharmacological potentials. In this specimen, we use Escherichia coli and Staphylococcus aureus. We focus on investigating the antibacterial properties of flavonoids extracted from various liquors against two common bacterial strains, so Staphylococcus and Escherichia coli. So both of these bacteria are significant in clinical and public health contexts due to their roles in various infections and diseases. The role of flavonoids in combating bacterial infections. So this study explores the potential therapeutic applications of flavonoids found in liquor with a focus on their natural antibacterial properties. So flavonoids, being potent bioactive compounds, may offer effective means to combat bacterial infection. In methodology, we use MIC, which is explained to be the method to determine the lowest concentration of the liquors that exhibits the visible growth of the bacteria. We also use MBC, the minimum bactericidal concentration, described to determine the lowest concentration of liquors that result in death of the bacteria. We also tested 3 specific wines: red wine, white wine, and rice wine. Flavonoid contents and its importance. So flavonoids such as anthocyanins found in berries and berry-like products like wine offer various health benefits including antioxidant, anti-inflammatory, antiviral, and anti-cancer properties. These flavonoids also is a key of interest, especially in combating bacterial strains like Escherichia coli and Staphylococcus aureus. One of the specific liquors to be analyzed is red wine, which emerged as a significant focus due to the promising antibacterial properties. The study acknowledged the variation in flavonoid contents among the different liquors, which could lead to identifying specific beverages with pronounced health benefits.
And here are the results. For this table, the MIC and MBC results of S aureus on red wine is discussed. So for the dilution on MIC, this column represents the various dilution tested to determine the lowest concentration at which growth of bacteria is inhibited. No growth indicates that the antimicrobial agent was effective at exhibiting bacterial growth in this case, and with growth indicates that the bacteria continued to grow, suggesting that this dilution were not sufficient to inhibit bacteria. For the dilution of MBC, this column represents the dilution tested to determine the lowest concentration at which bacteria are killed. No growth would indicate that the antimicrobial agent was effective at killing bacteria. The pattern is consistent with the MIC result with no bacterial growth seen to 1/2 to 1/128 dilution. However, starting from the 1/256 dilution with growth it was observed, indicating that these concentrations were not sufficient to kill bacteria. This means that the antimicrobial agent was effective at inhibiting and killing the bacteria at dilution up until 1/128. Beyond this, the bacteria demonstrate growth, indicating resistance or insufficient antimicrobial activity at lower concentration. This type of testing is crucial for determining the appropriate dosage of antimicrobial agent for testing infections. For the Table 2 on MIC and MBC result of SR use on white wine, according to the table, no microbial growth was observed at dilution from 1/2 to 1/256, which means that the agent is effective at inhibiting growth up to that dilution. At dilution of 1 to 512 and 1 to 1024, there is a microbial growth indicating that these concentration are not sufficient to inhibit the microorganism. For the MBC, also, according to the table, no microbial growth was observed also in 1/2 to 100, 1/256, which means that the agent is effective inhibiting growth, and 1/512 to 1/1024, indicating that this concentration is with growth and not sufficient to inhibit microorganism. In summary, the microbial agent is effective at inhibiting and killing the microorganism at the concentration of up until 1/256, and 1/512 is not effective at killing microorganism at this dilution. At 1/1024, it is no longer effective at indicating or killing the microorganism These results can help to determine the appropriate dosage of the antimicrobial agent for treating infections. For the Table 3 of MIC and MBC result of SR use and red wine, the result of both MIC and MBC indicate that the antimicrobial agent has an effective antibacterial range up until 1/256. Beyond this dilution, the agent ability to prevent the growth of the microorganism decrease, and at dilution of 1/512 and beyond, the agent fails to inhibit to kill the microorganism effectively.
MIC and MBC results of E. coli on red wine. The antimicrobial agent tested both on MIC and MBC does not inhibit or kill the microorganism at any of the dilutions provided in the test, suggesting that the microorganism is resistant to the agent or that the concentration used were not sufficient to have an effect. This table could be a part of a larger set of tests to find a concentration that is effective or to screen for more potent antimicrobial agents. Table 5, MIC and BC results of E. coli on white wine. So the dilution on MIC, the MIC is the lowest concentration of an antimicrobial that prevents visible growth of microorganism. In this table, no growth was observed at 1/2, 1/4, and 1/8 dilutions, indicating that the antimicrobial agent was effective in inhibiting the microorganism's growth at these concentrations. Starting from 1/16 dilutions and onwards, growth was observed, suggesting that these concentrations are not sufficient to inhibit the microorganisms. While on MBC, the MBC is the lowest concentration of an antimicrobial that results in the death of the microorganisms. Unlike MIC result, growth was observed at all dilutions, including the 1/2 concentration. This suggests that while the antimicrobial agent can be inhibited, the microorganism at lower dilutions, as seen in the MIC column, it does not kill the microorganism at any of the tested concentrations. And in conclusion, the antimicrobial agent has an inhibitory effect on the microorganism at higher concentration but does not exhibit a bactericidal effect at the concentrations tested. This could inform decisions on the use and efficacy of the antimicrobial agent for treatment purposes, considering the need for bacteriostatic versus bactericidal actions. MIC and MBC results of E. coli on rice wine. The table suggests that the antimicrobial agent is not effective against the tested microorganisms at the concentrations provided. This information is crucial for selecting an effective treatment regimen, as it indicates that either a higher concentration of the antimicrobial agent, a longer duration of treatment, or an alternative antimicrobial agent may be necessary to combat the particular microorganism.
The findings from the minimum inhibitory concentration or MIC and minimum bactericidal concentration, which is MBC, test conducted on Staphylococcus aureus and Escherichia coli with the different types of wine, namely the red wine, white wine, and rice wine. The results provide a comprehensive insight into the antimicrobial potential of this against the two bacterial species. The antimicrobial activity in red wine. For SRUs, the red wine demonstrated significant antimicrobial activity against SRUs with no bacterial growth observed in dilution up until 1/256. This suggests the presence of potent antimicrobial compounds in red wine possibly contributed to its higher phenolic content. For E. coli, red wine also showed consistent antibacterial growth across all dilution for E. coli, indicating a reduced antimicrobial effect on this antibacterium compared to SRUs. These variants may be due to the different structural and metabolic characteristics of E. coli and could make it more resistant to the compound present in red wine.
Antimicrobial activity in white wines. First one is Staph aureus. White wine effectively inhibit the growth of S. aureus up to a dilution of 1/256. Interestingly, growth was observed at 1/512 dilutions in MBC but not in MSC, suggesting bacteriostatic rather than bactericidal properties at certain conditions. While in E. coli, similar to red wine, white wine also exhibit limited antimicrobial activity against E. coli, with growth present in all dilution. This consistency further emphasized that we need to explore the different impacts of white components of various bacterial species. Antimicrobial activity in rice wine. First up is Staph aureus. Rice wine displayed antimicrobial activity, but growth was observed at a lower dilution, which is 1/512, compared to the red wine and white wine. This may reflect a lower concentration of antimicrobial compounds in rice wine or a different composition that is less effective against S. aureus. In E. coli, the rice wine, similar to other wines, showed limited efficacy against E. coli with growth present across all dilutions.
For the spectrophotometer result, the graph displays the absorbance of different wines, such as the red wine, white wine, and rice wine, along with the blank and the known or the positive control, which is measured at a wavelength of 550 nanometer. From this graph, we have observed that the red wine sample has the highest absorbance among the wine types. This indicates that it absorbs more at this wavelength compared to the others. This could be due to the highest concentration of color compounds in red wine. For the positive control sample, it has the highest overall absorbance because of this is the reference standard with known high absorbance properties. For interpretation, the red wine shows a lower absorbance compared to white and rice wines, which is consistent with its color. The rice wine absorbance is closer to the red wine, suggesting a similar concentration of absorbing compounds. For the blank or the expected lowest absorbance. This is usually the control sample to set the baseline for the measurement. This helps in understanding the actual absorbance due to the sample alone. This analysis helps in comparing the optical properties of these wines and can be useful in quality control, characterization, and understanding the concentration of color contributing compounds of these beverages.
Antibiotic disc diffusion test. The study investigated the antibacterial properties of different wines against Staphylococcus aureus and Escherichia coli. Using the antibiotic disc diffusion methods, the results were as follows. Red wine, 10 mm zone of inhibition, white wine, 6 mm zone of inhibition, and in rice wine, it's 6 mm zone of inhibition, which is found in Staphylococcus aureus. While in Escherichia coli, the results were as follows. Red wine, 0 mm zone of inhibition. White wine, 6 mm zone of inhibition. And rice wine, it's the same with red wine, which is 0 mm zone of inhibition.
For the antibacterial activity, these results indicates a variable antibacterial activity of the tested wines against the two bacterial strains. The differential antibacterial activity observed in this study suggests a selective efficacy of wine compounds against specific bacterial species such as the red wine, which is exhibit the highest antibacterial activity against the SRUs. This is due to its higher concentration of flavonoids, known to their antimicrobial properties. For the white wine and rice wine against SRUs, this could be attributed to the lower concentration of the bioactivity compounds. For the lack of inhibitory effect of red wine, rice wine on E. coli, this is because of the highest potential specificity in the antibacterial action of the compound which is present on the wines. Conversely, the moderate inhibitory effect of white wine on E. coli suggests that the presence of this different type of antibacterial agent is good for E. coli. For its conclusion, the comprehensive study we explored under the antimicrobial properties of red wine, white wine, and rice wine against two significant antibacterial strains of Staphylococcus aureus and E. coli. These employed or include MIC-MBC, spectrophotometry, and antibiotic disc or test finds or reveals that the red wine exhibits substantial antimicrobial activity against SRUs that likely contributed to its high phenolic content, yet showed less efficacy against E. coli. In contrast, the white wine displays moderate antibacterial effect against E. coli against both bacterial strains, while the rice wine indicated limited antimicrobial effectiveness.
The spectrophotometric analysis, which is measured at 550 nanometer, indicates that there is a notable difference in the phenolic composition of these wines. These are potentially correlated with their antimicrobial properties. These results have significant implications in fields like food preservation, where these wines extract could be leveraged as natural preservatives. And in medical applications, particularly for developing new treatments against antibiotic-resistant bacteria.
Based on the outcomes of this investigation into the antimicrobial properties of various wines, we recommend several steps to further capitalize on this finding. First, focus on research should aim at isolating and identifying the specific phenolic compounds in wines, particularly in red wine, responsible for their antimicrobial effects. Understanding the biochemical pathways through which the compound act on the bacteria can significantly contribute to the development of the new antimicrobial agent. Lastly, the wine industry should consider integrating these findings into their product processes. This could involve developing guidelines to optimize the antimicrobial properties of wines, which is potentially enhancing their utility in food preservation and safety. We also acknowledge that Centennial College Lab Prep Room for the cultures that they provide for us for this experiment. For the references, please refer to our list. Please here for more detailed information on the study and the literature of this research.
Thank you and happy holiday!